to impossible. So even the isolation of virus is next to impossible that we are trying to. Even we are just trying to dismantle viruses. So the amount of the genetic material, the RNA would be, it's insignificant. Even we cannot justify. So not possible because the size of virus is insignificant. So the nucleic acid, the size of the nucleic acid is also insignificant. So directly, if we are going to analyze the amount of the nature of the RNA, so for the analysis of the nature of the RNA, we must have the significant amount of this RNA. That is not possible after the isolation of this. That's why we are simply converting RNA into the DNA, and then further we are amplifying the amount of the DNA by cycle. Why rapid antigen test is not a confirmation test? Why? Because listen, the nature of antigenic protein, the nature of spike protein could be same. The nature of spike protein could be same in two different pathogens, right? But the nature of genetic material is always unique for a specific individual. My genetic material is different from the genetic material of mango plant. The mango plant genetic material is different from the bacteria. Bacterial genetic material is different from the common cold virus. Common cold virus, you just use your mind. Why we are talking about why we have classified viruses into so many heads? Why we have classified viruses into so many heads? Like one is the hepatitis virus, one is the rhinovirus, one is the coronavirus, one is the HIV virus. Why we are not talking about all viruses in the same language? Means commonly all are viruses. There must be some specification, and these specifications, there are so many specifications. These specifications are in the shape of viruses, in the mode of contamination, in the nature of the protein covering, and in the nature of nucleic acid. So possible, right? Two pathogens could be transmitted through the same medium. The two pathogens could have a same spike protein, right? But the two pathogens must have different in genetic material. That's why we are using this RNA for the confirmation, because the nature of RNA is always specific. The nature of RNA is always specific in the case of COVID-19. That's why it is used as a confirmation. Mamta, is common cold virus is less effective? ऐसा तो नहीं है. It depends upon the immunity. वो तो depend करता है. It depends upon the immunity of individual, ना? If you have a good immunity, common cold virus is less effective of. नहीं यार, it is not like that, because the nature of common cold is different, and coronavirus, the structure of coronavirus is different. The genetic material, the nature of genetic material of coronavirus is different, and the nature of genetic material and even the structure of common cold virus is different. Fine. So we cannot claim like both the viruses are same. Again, now the point is that why common cold is least effective against the coronavirus. That the reason could be what? Now we have seen this coronavirus in the recent period. Clear, and we don't have the body. First of all, our body start responding first of all against this coronavirus. But when we talk about this, common cold, so even since the childhood, we have seen this common cold infection. Right? Not only our generation, even father generation, grandfather generation, even we cannot imagine it to what extent, means how long we are facing this common cold infection. So then what happened over a period of time? Our ancestors have developed immunity against the common cold, and that immunity is being passed, or even we are also acquiring immunity against the common cold. But that is not possible in the case of coronavirus, because this virus has appeared recently. Right? Probably in our generation, if we are going to we are going to face this coronavirus forever in the ecosystem. So maybe after a couple of the years, this coronavirus will less effective to us because we have already developed immunity against the coronavirus. Again, if it is going to sustain forever, the next generation will be least prone to the coronavirus infection. Maybe generation after generation, even maybe after a couple of the generation, they are not going to be affected about the coronavirus because already this immunity is being transferred from me to my successive generation. Fine? So, coming to the next course of the discussion, and this is what? It's a cell. Coming to the next course of the discussion, and the cell is the basic unit of the life, and I told you earlier, people have yes ठीक है okay fine so now the cell is what? The cell is the basic unit of the life, and we have two categories of the cells. One is prokaryotic and the second is eukaryotic. The difference is what? We need to discuss only 5 minutes of the brief introduction about the cell. Otherwise, this is completely static or strict. But even we need to elaborate to some extent. This prokaryotic is evolutionary primitive, while it is evolutionary advanced. And the best example of the eukaryotic cell is what here? It's a plant cell, animal cell, and fungus. Right? And in these two cells here, this prokaryotic cell, less developed. This has less developed cell organelles, while it has well developed cell organelles. So the point is that what is the meaning of what we have mentioned here? If you just look at this slide, see here. Look here. If you just try to compare this slide, this is the good example of what we have mentioned. It's a good example of the prokaryotic cell. And we could see this cell in the case of bacteria. We could see here only the few structure inside, that is the nucleoid. It's a primitive nucleus with scattered DNA. Clear? So this structure is what? This structure is ill developed. Just have a look. You just have a look. Otherwise, explanation is not. Explanation to that extent is not required. Means in this structure, we could not see here nuclear membrane. We could not see here the structure like endoplasmic reticulum, Golgi complex, mitochondria, centrioles, etc. All are absent. So in the process of evolution, it was the first appeared cell. It was the first appeared cell, and we have mentioned the first appeared organism was the Monera, and the Monera was nothing but it was a bacteria, and the bacteria was nothing but it was the classical example of the prokaryotic cell. And the prokaryotic cell is a type of cell which is not actually cell. It's not actually a true cell, rather it is a prototype of cell, because this cell has not developed cell organelles like what we have mentioned here: endoplasmic reticulum, Golgi complex, mitochondria, centriole, and all. Right? But in contrast to this prokaryotic cell, if you just look at the eukaryotic cell, this is This is eukaryotic cell. It is advanced cell. Advanced. This we could see in the case of the plant, fungus, protista, anyone. And it has well-developed nuclear membrane. It has cell organelles like endoplasmic reticulum, mitochondria, Golgi bodies, centriole, plastids. So this is what? It's a well-defined. It is well-defined. It is well-defined. Fine? Like see here, ribosome for protein synthesis, centriole, which helps in cell division, Golgi bodies for the packaging of protein. It's a cytoplasm, that is the space. Nucleus, that is brain of cell. Mitochondria, power house. है ना? SER, smooth. RER, rough. Otherwise, both are endoplasmic reticulum. Yes, and the function of this endoplasmic reticulum is protein plus lipid formation. है ना? This lysosome suicide bag. Right, intermediate filaments for the skeleton of cell. Skeleton of cells. Fine, okay. So this is what? It's a well-defined structure. It's a well-defined structure. And these, this particular defined structure we could never see in the case of, आप देखो, we cannot see this structure in the case of prokaryotic cell. This is the major difference. So this cell is what? This cell is highly evolved cell. What we have mentioned as an evolved cell. It's an evolved cell. This is evolved cell. Why? Because it has so many cell organelles. And these cell organelles are well developed, and the intention of these cell organelles are what? Nothing but just to perform a specialized function. Just to perform a specialized function. Fine, okay. That is the general aspect of the eukaryotic cell. Yes? Golgi bodies, see, Meenakshi, it's a cell organelles. It's a cell organelles, and it is required for the packaging of the protein. And packaging of protein. What is the function of cytoplasm? Just now. Done? If it is done. If it is done, see, for all, it's a cell. It is the outermost part of the cell as a cell wall. That is only in plant cell. It is absent in animal cell. Yes? And the cell wall is made up of cellulose plus pectin. Yes, and just because of the cell wall, the plant cell cannot change shape. Just because of cell wall. Because cell wall is responsible for the hardness. Next to the cell wall, the next part is. The next part is here cell membrane, and it is present in all cells. In all cells. And this cell membrane is made up of, it's a tri-layer. One layer, two layer, three layer. Layer number 1, 2, 3. This first layer is protein. This second layer is lipid. Lipid means fat. And this third layer is protein. So the cell wall is tri-layered in nature. Okay? Then this centrally located part is nucleus, and the nucleus consists of genetic material, which is DNA, RNA, or we also call it as a gene. Fine, right? And then the space between nucleus and cell membrane. This is what? It's a space. And this space is cytoplasm. This space is cytoplasm. And this cytoplasm consists of cell organelles, like what we have mentioned here: endoplasmic reticulum, lysosome, plastids, mitochondria, etc., etc. Fine. So the function of cytoplasm is what? Cytoplasm is space. Cytoplasm is a space, peripheral space. That space is a place for all cell organelles. All cell organelles. Right? So except the nucleus, the rest of the cell organelles are lying where exactly? In cytoplasm. Right? Fine? Yes? Done? Done? So listen, if you see here, the basic difference between the plant and animal cell, only few differences are required. Both are eukaryotes. Both are eukaryotic cells. One is plant cell and the second is animal cell. Fine? So almost we can see the structure are similar in plant and animal cell, but only the difference is what? In the case of animal cell, no cell wall, cell wall is present. In the case of plant cell, you see one large vacuole, one large vacuole is space. But in the case of plant cell, we can see here, a number of small vacuoles like this. Small vacuoles. Fine. And the third and the most important part, in the case of plant cell, there's a plastids, and the plastid is involved in food synthesis plus food storage. Means photosynthesis. Photosynthesis. Right? Photosynthesis. Okay? Done? So here, this is what it's the basic structure of the cell. That is required just for the information. Otherwise, in so many years, after 20 years, they asked one question from this particular section. But even this basic discussion is required because we need elaboration to some extent. So, see here. In cell, nucleus consists of gene, means genetic material. Nucleus consists of gene or genetic material. And the genetic material is responsible for the determination of characters. But apart from the nucleus, mitochondria and chloroplast. Chloroplast is a type of plastid. These two consist of their own gene. Right? These consist of their own gene. So, these two cell organelles are partially controlled by nuclear gene, partially controlled by nuclear gene, and partially controlled by their own gene. Partially controlled by nuclear gene and partially controlled by their own gene, and that is the logic behind these two cell organelles are called as semi-autonomous cell organelles. Semi-autonomous cell organelles. Fine, okay? Right? So see, there is a question like, apart from the nucleus, chloroplast and the mitochondria are the two cell organelles consist of their own gene, right? Fine. And here I need to elaborate this to some extent. This is nucleus. And this nucleus consists of 20,000 to 25,000 gene sequences. The nucleus of the one cell consists of 20. Just taking a 30-second break. I'm not going outside. Just there was a good question from the Vidisha, which I missed. Vidisha, if you can hear me. Now this question is very common nowadays, and I'm just taking a logical questions. Suppose someone has affected by corona, like just taking the best example, it would mean that the antigen has already entered in the body. Obviously, okay? Means my antigenic spike protein means I got infected to the spike protein of coronavirus. Fine. So against this, my body is already responded in the term of antibodies. Okay, fine. Okay. So then what happened? See, after 28 days means since the infection to 28 days, clear? My body will respond at the maximum. Means my body will respond at the maximum means the amount of the antibody will be maximum after 28 days. After 20 days, and the position of the antibody is where exactly in the plasma. Fine. But this antibody is not in everlasting position. It is also going to be diminished. It is also once I am going to be clear through infection, this antibody is going to be diminished. Means the amount of the antibody is going to be subside. Is the logic behind why we are talking about the plasma therapy nowadays. We are asking for the plasma donation for the only those patients we have already recovered, but not more than 3 months, because that is the period that we have a maximum amount of the antibody against coronavirus, because after the 3 months, the amount of the antibody is going to be subside. Right? So once the amount of the antibody is going to be subside, clear, means we will have a trace of the antibody, and after 3 or maybe 6 months or after the 1 year, clear. And even if the virus is going to affect with the new strain, so definitely that antibody is not effective. It is not effective, and again I am going to be infected. Right? That is the reason then I infected twice. So it is not possible. It is not like that. If someone is infected to the coronavirus, so he or she is not going to be infected in the future. So the chance is less, but the infection is right. Infection could be possible, and even it is not assured that it is not assurance that if someone is infected to the corona, so vaccination is not required. And vaccination is the medicated dose. Vaccination is the medicated dose, right? Just to protect you against the corona infection in the future, right? So it is not like that. Once you recovered, right? So the corona, once you recovered against the corona, so you are not supposed to take vaccination. Vaccination is always preventive measure, just to check the further infection of the corona. We need this preventive. It's not corrective, it's not curative, rather it's a preventive measure. Fine. So Vidisha, that question, it was the relevant question and I missed and I replied. So again I'm coming back to this discussion. Rahul, how animal cell can make their own food home? Rahul, can you hear me? आप बताओ ना. See, when we talk about are we able to photosynthesize? We are banking upon Yes, Siddhant. Yes. Siddhant Chaudhary. It's a logical answer, but the recovery will be faster next time as the body will recognize the virus as, see, obviously, when someone is going to be infected through the virus again and again, so recovery, the pace of recovery would be faster, right? That is the correct explanation. Fine. So, ये बताओ, I was just talking to Rahul. Are we able to photosynthesize? We are banking upon the food. See, we are heterotrophic in mode of nutrition. Plants are autotrophic. Plants have chloroplast. So plants are able to photosynthesize. But for that, when we talk about the animal, any category of the animal, whether we talk about the human or even, we are banking upon the food sources for the food. We are banking upon the sources of the food, right? So we are cultivating wheat, rice and all. We are not, it is not like that once we feel hungry, so we need to go to the, we need to go to the sunlight and then we start photosynthesizing. That is not possible in the case of the human. So in our case, the mode of nutrition is what? We are banking upon the other source for the food. But in the case of plants are not banking upon the other sources. Why? Because plants are blessed with the chloroplast and the chloroplast has a ability to photosynthesize, right? So just hold it. We are going to explain this photosynthesis and the artificial photosynthesis, but that is the next part of the explanation, right? Okay. So this nucleus has 20 to 25,000 of the gene sequences and the 50%, and here the source of the gene sequences are what? The 50% of the gene we are going to get from the father. 50% from the father and 50% from the mother. Father, sperm and the mother, egg. Clear? So this nuclear genetic material is both maternal and paternal. But here the cell has mitochondria. And the mitochondria has gene. And the mitochondria has only 37 gene sequences. And the source of this mitochondrial gene is only mother, maternal. Mother means female. Right? The source of mitochondrial gene is only mother. Means I have mitochondrial gene in my cells, and the source of my mitochondrial gene is what? My mother. My mother has mitochondrial gene, and the source of my mitochondrial gene is my maternal grandmother. Right? So the mitochondrial DNA inheritance is female to female. It is only through the mother. While the nuclear genetic material is both through mother and the father. Alright? And see here, now the amount of the nuclear gene is what? It's a 20 to 25, while the mitochondrial DNA is what? It's a 37 gene sequences. Means the more than 99.9% of genetic material is nuclear in nature, and it is less than 1.1% of genetic material which is mitochondrial in nature. Fine? But okay, so see what happened in the process of fertilization. In the process of fertilization, father is the source of sperm. Sperm is what? It's a cell. Like it's a eukaryotic cell. Like it has a nucleus and it also has mitochondria. And it's a cell. So it has all cell organelles. Mother, egg. Egg has nucleus and egg has also mitochondria. It's a cell. It's also eukaryotic cell. So in the process of fertilization, that is syngamy. What happened? This is sperm, this is the egg with this and this. And this sperm will try to penetrate this sperm will try to penetrate egg, and then this sperm will discharge only nucleus part. Only nucleus, and then this will turn into zygote, and this zygote will have nucleus from both mother and father, and this zygote will have mitochondria of the same female. Mitochondrial gene of the mother, same female. Fine? And here, this nuclear gene is 20 to 25,000 gene sequences, while the mitochondrial gene is 37 gene sequences. Right? So UPSC is not going to ask about the mitochondria as a source of, as a powerhouse of the cell. Clear? So see what happened. If this mitochondrial gene is faulty, if this mitochondrial gene is defected, if this is what, it's a zygote, it's a zygote, it's a zygote, and if mitochondrial gene is defective, baby will suffer from brain disorder. Baby will suffer from brain disorder. So for that, and you must be knowing about more than 80 to 90% of the brain development is done in the 9 months of the gestation. Right? We born with, the newly born child has more than 80% of the developed brain, and the rest of the 10 to 20% of the brain development is over by the age of 8 to 10 years. Maximum by the age of 8 to 10 years, brain development is done, and once the brain development is over, physical development starts. So see, physical development starts like the sudden growth, fine, the appearance of the facial hair, right, the onset of the sexuality, the development of the breast in female, menstruation. This starts after the age of 10 to 12 years, once the brain development is over. Right? So newly born child has already developed, has already 80 to 90% of the developed brain and 10% of the physical development. At the time of the birth, we have 10% of the physical development and 80 to 90% of the developed brain. And then after the birth, the rest of the 10 to 20% of the brain development is going to be done by the age of 8 to 10 years. Until that period, the physical development is slow. And once we attain 100% of the brain development, fine, so the physical development is going to be faster. Right? So suddenly you could see, in the short span of the 3 to 4 to 5 years, sudden elongation of individual, sudden expression of the sexual ability, right? Because it indicates that if the mental development is done, and now if the mental development is done, means physical development starts. Right? So here, for the development of this brain, here the mitochondrial DNA of the mother plays the most important role. And if the mitochondrial DNA of mother is faulty, in that case, there could be the chance of the abortion of the baby, because this baby is not going to be developed because of the brain disorder. And before coming to this, and just to handle this faulty mitochondrial gene, just to handle this faulty mitochondrial gene, in 2014, a new technique was approved in UK, and this is what? It's a MTR, mitochondrial gene replacement. Clear? Mitochondrial gene replacement, and here the product is named as triparental baby. Triparental baby means the baby has, triparental means baby has 3 gene. Means baby has gene from 3 different sources. Clear? Fine. So see here, just look at the case study. This is the best example of what the people are asking. This is the case study. Sharon Bernardi, she knows all too well the pain of living with the mitochondrial disease. A 47 years old from the Sundarland has lost 8 children to the genetic conditions. 6 of them within hours of the birth, and thus she was forced to abort at 21st weeks because of the severe defect, and her only surviving son Edward died in 2011, aged 21, after surviving from Leigh disease, a rare hereditary condition affecting the brain and nervous system. And this is because of faulty mitochondrial DNA. Right? Yes? So just to handle this mitochondrial gene replacement technology was approved in UK, and we named this product as a triparental baby. Clear? So see, this is what? It's a mitochondria. And this is what? It's a nucleus of the cell. And so this nucleus controls the action of all the cell organelles. But I told you this mitochondria has mitochondrial DNA that consists of 37 gene sequences, right? So these gene sequences controls the action of mitochondria, as well as mitochondria itself is controlled by nuclear DNA, and that is the reason behind why mitochondria is called as the why mitochondria is called as semi-autonomous body, because partially controlled by nuclear DNA and partially controlled by its own DNA. Fine. So now here the intention is what? Just to replace this faulty mitochondrial DNA. Clear? Just to replace this faulty mitochondrial DNA, we adopted this technique as a MTR that was approved in 2014. So what actually is done in this particular technique? See here. See. Just look at this slide. This is father's sperm. Sperm is always nucleus donor. This is the mother egg. Nucleus, and this mother has faulty mitochondrial DNA. Okay? Clear? And this is the donor egg. That we can get it from the egg bank. Fine. And from the donor egg, we need to discard this nucleus part. Fine. We need to discard this nucleus part, and then this is what? Donor egg with mitochondria. Healthy mitochondria. Fine? And we need to take nucleus from here outside and then we'll insert here inside. Now this is what? It's a healthy egg. And this egg has nucleus from the mother and mitochondrial DNA from the donor's egg. And it is allowed to fertilize. This is allowed to fertilize. This is sperm with the father. It is allowed to fertilize the sperm of the father. Fine. So now this baby will born, and this baby is going to have gene from 3 different sources. One source is this, from the father is sperm, and here is what? It's a nuclear gene. This is the mother egg. This is what? It's a nuclear gene, and this is the donor's egg. And this donor egg is what? It's a mitochondrial gene. So this baby is going to have gene from 3 different sources. Donor's egg mitochondrial gene, mother's egg nuclear gene, and the father's egg nuclear gene. And we named this baby as a tri-parental baby. Fine? Okay? Fine? So how this could be done? Listen, this entire discussion, this could be done in 2 ways. One, the entire technique is done. One is before fertilization, and the second is after fertilization. So listen, let me explain, let me elaborate. This is the first one. It is before fertilization. This is called as maternal spindle transfer. Hold your question, Namrata, please. I am just coming to you. Let me complete. I am going to answer all questions. Just hold your question. Don't be panic. Second is after fertilization is called as pronuclear transfer. If it is done before fertilization, this is called as maternal spindle transfer, and if it is done after fertilization, it is called as the pronuclear transfer. So what actually is done in maternal spindle transfer? See here, this is the patient egg. Patient means mother egg. Which has faulty mitochondrial DNA. And this is the donor's egg, which has healthy mitochondrial DNA. Yes? So you could see here, you could easily see here, this mitochondrial DNA is faulty. we need to take this nucleus outside. We have simply taken this nucleus part, means rejected this entire part. The entire part is rejected. We have simply taken this nuclear part. I think this color is good. See, we have simply removed this nuclear part, and in the donor's egg, see, this nuclear part is there. We have simply cleared this nuclear genetic material, removed. So this has a cytoplasm. You know, this is what it has a cytoplasm cell with mitochondrial DNA, and it is being inserted with the nucleus of this. So an artificial cell is made by inserting nucleus of biological mother into the cytoplasm, nucleus of the biological mother into the cytoplasm of donor's egg. Fine. So now this egg has two different genes. One is mother nucleus and the second is donor mitochondrial DNA, and then it is allowed to fertilize. It is allowed to fertilize with sperm like this. So this fertilization leads to zygote, then it leads to embryo, and then it leads to baby. Fine? Okay? Done? While in the case of after fertilization, see here, this is the patient egg fertilized with the sperm, and here is what? It's a zygote. It's a zygote with faulty mitochondrial gene. Right? It is zygote with the faulty mitochondrial gene. Fine. See here, it is a donor's egg with sperm fertilized. This is the healthy. Zygote with healthy mitochondrial gene. And here what we have done, we have simply cleared off nuclear genetic material of this. See, it is being removed. So what we have a cell with mitochondria, and from here we have simply taken a nucleus part outside, which we have inserted here. Means we have cleared cytoplasmic part, cleared cytoplasm with mitochondria gene, and only done nucleus. So this cell, see, this is already been inserted. So this is what? It's a healthy zygote, which leads to embryo, and this leads to baby. Yes? Fine? Fine. So again repeating in linguistic manner, in the maternal spin transfer, what happened? Before the fertilization, the mitochondrial defective mitochondrial gene of mother egg is fixed by transferring nucleus of biological mother into the cytoplasm, egg cytoplasm with the healthy mitochondrial DNA of the donor's egg, which is followed by fertilization process. Means the cytoplasm with the faulty mitochondrial DNA of biological mother is removed and the nucleus is taken out, and that nucleus is transferred to the donor's egg with healthy cytoplasm, with healthy mitochondrial gene in cytoplasm, and then followed by fertilization. While in the case of pronuclear transfer, we are performing fertilization, two different fertilization independently. One is fertilization between the healthy egg and the healthy sperm, result into the zygote, and the second is fertilization between the healthy sperm and defected egg with the mitochondrial gene. Right, and then the nucleus and the zygote results. Then the nucleus of this biological product with the healthy mitochondrial gene is taken outside, and then it is transferred to the cytoplasmic part of the zygote, which is the product of healthy egg with the healthy sperm. Clear? After removing the nucleus of that zygote, which is the product of healthy sperm and the healthy egg, and then followed by the fertilization. Clear? Followed by fertilization. And the result is what? Result is now this baby this baby will develop with brain efficiency as a normal baby. Fine. So that was the question. I think this was the question. Last day, let me show you the question. See, this was the question in 2020. See, aap dekho na. This was the question in 2020. In the context of the recent advance in human reproductive system, the pronuclear transfer is used for. This was the question in 2020. Prevention of mitochondrial disease in offspring. It's a kind of gene therapy. We have so many approaches in the gene therapy, and it is one of the examples. Gene therapy is a wider term, but this is one of the classical example of gene therapy of what we have done, and the intention is just to fix the faulty mitochondrial DNA, right? And you see, there is one more question from this entire section, means what we have discussed so far in the today's lecture. The general difference between the plant and animal cell. The plant cell has low cell wall, while the animal cell does not. Correct. The plant cell do not have plasma membrane, unlike the animal cell. Galat. The mature plant cell has a large vacuole, while the animal cell has many small vacuoles. Right, you see here. So this was a question 2020. I will show you the question so that you feel comfortable how we are supposed to do like.